Today, we're going to talk about the pre-released scientific article for the International Biology A-Level that is linked with question 8 of Unit 5. Your teacher will tell you to read this article several times in order to understand. However, time is a precious commodity, especially with our jam-packed schedules filled with classes, work, and life's curveballs. That's where this strategy comes in handy. Here's the game changer: read the article once, and then let this video serve as your study companion, where I'll read the article aloud to you. By doing this, you not only comprehend and retain the article's content, but also ensure that on exam day, you'll effortlessly locate the answers without sifting through the article again. Whether you're on your daily jog, commuting, or simply taking a leisurely stroll, hit that play button repeatedly. So, get ready to maximize your study efficiency, and let's dive right in. Pre-released scientific article for IAL Biology. Article 1. One species of whitefly, an aphid-like insect, has incorporated a portion of plant DNA into its genome that protects it from leaf toxins. It seems to be the first known example of so-called horizontal gene transfer between a plant and insect in which the transferred genetic material performs a useful function. While sequencing the genome of the silverleaf whitefly, Bemisia tabasi, Ted Tillings at the University of Neuchâtel in Switzerland and his colleagues discovered a gene known as BTP-MAT1, which is found in plants but never previously seen in insects. This gene may have an important function in plants. The plants generate toxins to defend themselves from attack by animals. The team suspects that the BTP-MAT1 gene may help plants store these toxins in a harmless form so the plants don't poison themselves. Similarly, the gene may help the whitefly avoid being poisoned when it eats the plant. Tillings says the gene transfer event occurred between 35 million and 80 million years ago, when the silverleaf whitefly and other whitefly species that lack the gene split from a common ancestor. The gene transfer event may have involved viruses that cause disease in plants and are transmitted via the whiteflies. Some DNA from a plant may have been taken up by a virus, transmitted to the whiteflies, and then subsequently assimilated into the insect's genomes. Some viruses basically incorporate their own genome into the cells of their hosts, says Tillings. The research suggests that the extent to which horizontal gene transfer occurs in nature is probably underestimated, says Caitlin Burt at the Australian National University in Canberra. What this shows is that where there's a really strong pressure for survival on an organism, it can actually borrow genetic information that helps it do that from other organisms, says Burt. The researchers demonstrated the function of BTP-MAT1 gene in whiteflies by selectively interfering with the gene using small molecules of RNA. Disrupting the gene's function made the whiteflies susceptible to compounds known as phenolic glycosides that are present in tomato plants. After feeding on tomato plants that had been genetically modified to produce the RNA molecules, all whiteflies subsequently died. This demonstrates a mechanism that we could use in engineering crops to basically target plant pests and target the resistance of crops to plant pests, says Burt, although she points out that horizontal gene transfer may then allow the pests to evolve resistance to our genetic engineering. Article 2. Glowfish are fluorescent transgenic zebrafish. These are illegal in many countries. Glowfish are zebrafish containing a DS-red gene from a sea coral that makes the fish fluorescent under ultraviolet light. Zebrafish are normally silver and black. There are several versions of the DS-red gene, each resulting in a different colored phenotype, from fluorescent yellow to fluorescent red. As it is illegal to import these transgenic fish into many countries, technology is used to detect them. A PCR-based method has been developed to detect transgenic zebrafish harboring the gene, DS-red, coding for the red fluorescent protein, originally isolated from the marine sponge Discosoma striata. 2 types of PCR have been performed. PCR to detect amplifiable genomic zebrafish DNA was checked using primer specific for the zebrafish parvalbumin gene. PCR with primers to specifically amplify the DS-red gene. In both PCR systems, genomic DNA isolated from wild-type zebrafish was used as a control template. In the second PCR system, the plasmid DS-Red2N1 was used as a positive control. Applying this method to several specimens of presumed glowfish from traders in the Netherlands and Germany revealed the presence of transgenic fish. In addition, a rapid method for screening zebrafish suspected to be genetically modified has been developed by measuring the fluorescence of water-soluble protein. Article 3. Red and yellow and at least 70 other colors. A genetically engineered fish has skin cells in all the colors of the rainbow and then some. Its beauty is more than skin deep though. The huge variation in color could be used to track individual cells as they develop, move, and regenerate. The skin-bow zebrafish was created using a gene that codes for red, blue, and green fluorescent proteins, although only one color is produced at a time. Ken Poss at Duke University in Durham, North Carolina, and his colleagues injected this gene into single-cell fish embryos. In one particular embryo, this gene became incorporated into part of the genome that patterns skin cells. As an adult, this fish looked reddish in color, but when the team shone a UV light on its skin, it lit up in Technicolor. We didn't know these patterns would develop just in the skin, says Poss. When you make genetically engineered animals, you can't fully predict the outcome. Poss thinks that each of the fish's skin cells has about 100 copies of the gene. Whether each gene creates a red, green, or blue protein is entirely random, he says. One cell might have 80 red, 10 green, and 10 blue proteins, for example, says Poss. Each cell could have one of about 5,000 possible color combinations, although the resolution of Poss's microscope only lets him distinguish 70 different colors among the cells. Regeneration and repair. The fish and its offspring can be used to track how skin cells move to regenerate tissue and repair injuries, a task normally made difficult by the fact that individual skin cells look so similar to each other. In one experiment, Poss's team took snapshots from one patch of skin, twice a day for 20 days. All the images were fed into a computer, with software that can identify and track each skin cell based on its color. This allowed the team to work out that the entire population of the fish's skin cells turns over every 20 days or so, and that each cell spends about 8 days on the surface of the skin before it wears off. The team also watched how the fish's skin responds to injury. The group snipped off part of a fin, an injury that zebrafish can usually repair. Skin cells in the surrounding. This approach lets us image cell dynamics in a live animal, says Poss. He hopes that the skin-bow fish can be used to reveal more about how tissue regeneration occurs, a process that is still mysterious, he says. Christine Pollard, who studies wound healing at the University of Leicester, UK, agrees. I can imagine that this group will be able to explore skin biology on a new level, she says. I look forward to seeing more research on skin-bow. At the end of the article you can find 3 references, one for each section. If you are looking for notes, study guides, or potential questions in association with this article, you will be happy to know that by visiting www.tes.com you will be able to find any resource you wish for. We have our own online shop, hosted on the TES website. It is called Biology A-Level Tutor, and there you can find a comprehensive set of notes with more than 119 potential questions with their answers based on this article. Our shop is your one-stop destination for educational excellence. Visit today and explore the resources available. If you found this video helpful, don't forget to give it a thumbs up, share to other students, and subscribe to our channel for more valuable study tips and strategies. Thank you so much for tuning in, and best of luck with your studies and exams. Keep learning, keep growing, and I'll see you in the next video.